So I'm here today just to introduce Manuel. So it's my real pleasure to introduce Manuel Kaulich. He's a really leading expert in gene editing and CRISPR technologies. Manuel has trained at the Max Planck Institute and the Biozentrum in Basel and University of California, San Diego. And really his research has uncovered key mechanisms in cell cycle regulation and chromosome segregation. Now, as a group leader, as a Goethe at the Goethe Institute, Goethe University in Frankfurt, he is also a head of the gene editing core facility there. So, over the course of his kind of career, he has developed innovative CRISPR methods and protocols that really advance not only basic but also applied research. He is also a co-founder of Vivian, which is a company that provides new tools to make CRISPR screening more effective and accessible. So I think you'll all agree that he's a really great speaker to participate in this course. So without further ado, I just want to introduce Manuel and let him get on with his fascinating talk.
Thanks a lot, Panos. I can't remember having been introduced so kindly, so it's been a while, so thanks a lot for these kind words. And it's a real pleasure to have that opportunity actually and share obviously the basic concepts of today's topic, which is called CRISPR screening, but maybe also some of the things that we learned over the past couple of years that you don't read in papers, which you only learn by actually doing this. And I try to find a good balance to mix basically with things that that are general, but also spread in some of the information that, as I said, that you won't read, and but we think it's critical. Yeah. So I would like to start with a question, which is actually, why do we need to screen in the first place? Because people have sequenced all the genomes all the time. We know GWAS, we have lots of variants, and why do we need to screen? And I think I like this analogy of when you look at the blueprint of, for example, a car, right? You have like a gazillion parts. And that is basically what you would get when you sequence. When you sequence a human genome, a bacterial genome, it doesn't matter. You would get sort of like a map, right? You get a map of the parts, but you don't actually know what these parts are good for. So basically, what's the function? So for example, you may be able to detect the wheel, right? And you may be able to detect the door, but you don't actually know what that function is. So you would know, okay, this looks like a door, but I mean, what's the function of a door? This is a wheel, and you wouldn't know because simply sequence is insufficient to tell you about the function of that sequence. And the best case example that I could come up with in this particular case is, for example, imagine now you're just sequencing and you find this wheel. But the wheel, under normal circumstances, I would argue, is non-essential, because if the car stands, you can take away the wheel and nothing will happen, right? But what happens, and this is exactly the case, right? If the car stands, the wheel is not essential. So basically, for a non-proliferative cell, you would not be depending on a wheel. But what happens if the car drives, right? If it drives, it proliferates. Now, all in the sudden, the wheel becomes essential because you would be nicking to one side. And it's even more important. So if you start to turn the car, right, you would realize, okay, that sequence apparently that I perturbed there, it has to do with not only driving forward, but also making turns. So you can infer function by perturbing the wheel, right? And you can continue this by, for example, with a tuned car, you can take away parts, and then the analogy in this case would be cancer. So the point that I want to make here is that sequence does not tell you about function unless you know something about the sequence beforehand, right? And this is why now GWAS studies, for example, or variant calling, they basically start to perturb these variants and see what is actually the function of them. So they're using that. So the point I want to make, perturbation reveals function. And the best case example here is, imagine removing a wheel from a car that is driving or turning. And that will tell you basically, or will allow you to infer that function from the phenotype that you're observing, right? And the best example in the literature out there when it comes to CRISPR screening is actually the identification of core essential genes. So basically genes that when you interfere with their function by means of their perturbation with CRISPR, a cell becomes non-proliferative. So that means the function of that gene is required for the cell to be fit or to proliferate. I'm just citing here a study from a close friend, Trevor Hart. And what they have done is they have looked at many different cell lines mutant or wild type from RPE cells, HTTs, A375, and so on. And what they did is they applied genome-wide screening and asked which of the genes are causing these cells to perform less fit, right? Less well. So which genes are dropping out? And we will come to how this is calculated a little later. And then depending on the cell type they're looking at, they found in the order of 1,500 to 2,000 genes that were dropping out. And then basically the sum of those, or the overlap of those, this is what we today consider core essential fitness genes, right? So independent of the context that you are investigating the function of those genes, they will be essential. And those are, for example, genes that are strongly associated with the translation, for example, so ribosomal genes, or the proteasome, protein degradation, or transcription, like RNA polymerases, the mediator complex. All of these types of things that are super essential for any cell to survive and to proliferate, those will be core essential, right? And so in this case, genes required for cell proliferation, that's our driving car analogy, they can be identified in that. Okay, so now we need to perturb. Okay, so we need to screen. So how do we screen? And I'm just starting again with the basic overview that probably many of you have seen already. And what I would like to do now is to go step by step through the individual components of the screen and basically tell you, share with you a little knowledge that we have when it comes to these aspects. And again, I divided this here, as I said. So we'll talk a little bit about libraries. We talk about the viruses that you will need. General, the screening setup. So what sort of like at this point, I will provide you with what I consider a checklist that everyone should go through before starting a screen. Then we'll talk a little bit about phenotypes. So what type of different phenotypes can you screen for? There are simple phenotypes, there are complex phenotypes. We'll briefly touch upon NGS and how you turn your sequencing data basically into read counts. And then I know that this has been a topic already for you. We will touch a little bit upon quality control of your sequencing, but also what means of data analysis out there and what type of things you should be aware of and not to generate false positive hits. Okay, so let's start with the library. There are, generally speaking, 3 different types of libraries that more or less all libraries fall into. The first one, those are just monogenetic libraries, single guide libraries. That means you end up with 1 guide per cell, right? And those library formats, they're application agnostic. So that means that can be knockout, can be activation, can be repression, base editing, for you name it. But the concept is single library means 1 edit per cell. The second case that I want to make is fixed pair. So that means you have predetermined combinations of guides in a single cell. Yeah, and this can be multiple guides. This can be a combinations of 2 is probably the best example here, where you have in a single cell you have the combination of A with B, and in the cell next to it you have C to D, and in the next cell you have E to F. So you basically have predetermined combination, and this can become handy, for example, for analyzing paralog genes. Right? Those are basically genes that have the same ancestor, that have very similar function. And if you deplete one of them, you don't really see a phenotype. So you need to deplete both or all paralogs in order to see a phenotype. And this can be done with such reagents. And the third category that I want to point out is what we consider multiplex libraries. So that means you also have multiple guides in a cell, but you don't have predetermined combinations, but instead you have all by all combinations. So imagine you have 100 genes on one side and 100 genes on the other side, then your total library diversity would be 100 times 100, so 10,000. So your library size is 10,000 elements. And you don't know which of those combinations end up in a single cell because you're testing all of them at the same time. Yeah. So single library, the size of a single library is obviously the number of guides that you have. For fixed pair, it's the number of predetermined combinations. And for multiplex, the size is basically the square of the genes you want to investigate. Yeah. And so obviously multiplex experiments, they scale very badly because imagine you want to investigate 1,000 genes. That's 1,000 square minus 1,000. That's a huge number. That's right. That's a million combinations. Actually, it's 999,000. So this is, you don't have to read all of this. Just listen to me. I want to put up here a complicated slide because there are a gazillion different ways in how you can generate these libraries. All right. You don't need to look at this. I know it's recorded, so you can look at this later. And I just mentioned here like the most used ways of generating these reagents. And those are divided into basic cloning procedure, which is basically Golden Gate, Gibson assembly, restriction ligation, but also infusion Lick dependent ligation, right? So these are all of these four here, what they have in common is that when you get your oligo pool, when you want to make, basically when you want to clone your libraries, that you have to PCR amplify the oligo pool and then drop it in either with a ligase or with a recombinase or with homology sites into a target backbone, right? Like conventional cloning. This is what they have in common. The technology that I want to basically not point out, that's the wrong word, but that I want to highlight a little bit is actually a PCR and cloning independent technology that we actually developed. And that's called CRISPR, so it stands for covalently closed circular synthesized guide RNA. And it doesn't really matter, I don't want to go into details how this works, but in essence, what we do is we avoid the PCR amplification of the oligo pool. And that has huge consequences for the distribution of your library. And I will explain what this means later on a little bit. Bear with me and know that for this particular technology, you end up with extreme uniform libraries. So that means that the representation of guides within your library is very similar. So basically 1 to 1 to 1. Each guide is more or less as often present as any other guide in the library, which is not the case for the ligation dependent libraries, because again, you have a PCR and you do cloning. So you end up basically on skewing your oligo pool into a more positive or more negative guide RNAs. So now let's say, and that is again just highlighting here. So when you have your when you design your CRISPR library, you order this oligo pool, you PCR it, or you do 3CS cloning, and then you drop it basically into a lentiviral backbone. And the next logical step here would then to use this library and turn it into viral particles, because that's important, because we want to make sure that in the experiment later on, each cell only receives a single element. After the worst case that can happen is that a cell receives multiple elements, and then our phenotype gets diluted, because then we will not be able to say, all right, that cell had that phenotype, because it's a multiplex problem. We cannot entangle this. So that's a very essential part here. So just basically the basic protocol on how we make this. So depending on the generation of lentiviral particles that we use, and we will come to what this means, is you end up with probably like 2 or 3 plasmids. So first, plasmid is called the transfer plasmid, and this is basically the plasmid that encodes your guide RNA. Yeah. Then you have an envelope plasmid and another packaging plasmid, and these 2 plasmids, they encode the elements the cell need to express so that the viral particle can be generated and expressed. So those are envelopes, those are RTs, and so on. And basically what you do is you take those 3 plasmids and you transfect it into HEC293 T cells. Don't do screens in HEC293 T cells. I just want to point this out right away. This is the worst thing you can do. I always criticize this when I'm a reviewer, because these cells, they can do one thing only, and that's protein expression for lentiviral production. And that's the only thing. So, but they do that really great. So you take those 3 plasmids, you transfect it into HEC, and what they do what they do best, protein expression. And in this case, that will lead to lentivirus generation in the supernatant. Yeah, so then you collect the supernatant basically, which contains all your lenti, and then with an intermediate step where we still have to determine the titer, on the next slide we can then use this for transduction. So because that's important for some of the applications, I just want to highlight here on the right-hand side the difference between 2nd and 3rd generation lentiviral plasmids. So, and this relates to what I said, how many plasmids you will need to produce these libraries. So 3 plasmids, that relates to the 2nd generation. So you have a transfer plasmid, you have packaging plasmid, which contains GAG, POL, TAT, and so on and so on. And you have an envelope plasmid, which contains this envelope protein that forms actually the lenti. And the difference to a 3rd generation is simply that this packaging plasmid from the 2nd generation has been split into 2, and this set element has been removed, and that makes it a little safer, but and not sort of like recombination stuff, but it doesn't really matter. So both of these systems are very safe and easy to handle. And if you use a third generation plasmid transfer vector, it can be used with both second and third generation packaging system. So it's interchangeable. Yeah? But I just want to say this. All right. So then once you have collected that virus, obviously what you need to do is you need to know how many viral particles have you actually generated. So, and that's important because later on, as I said, you need to make sure that each cell receives a single element. Yeah. And the way this works is by serial dilution. So you plate your target cell in which you would like to do the screen. And then you, in a stable cell number, let's say you always put like 20,000 cells into one of those 12-volt plates. And then you generate a serial dilution of the lentiviral particles, right? So you put in maybe 20 microliter into the first tube, into 1 ml. So then you mix it and you take another 20 into the second. You mix it, another 20. You mix it, another 20. And so you end up with a dilution series. And now what you do is you take, again, from this dilution series, you take some, maybe 20 microliter, and you add this onto your cell. And now after you added your cells, you apply a selection pressure. So in this case, the lentivirus, that your transfer plasmid not only encodes for the guide RNA, but also, for example, for an antibiotic resistance. So once you transduce, after the next day or the 2 days after, you change the media, and then you apply that selection pressure, for example, puromycin, blastcysteine, or hygromycin, and stuff like this. So you're basically killing everything that didn't take a bite. Right? And then you wait a few days, and then there are different ways of doing this. You can either you do a crystal violet staining, or you can just go to the microscope and count the colonies per plate, right? Or you go to the fax machine, because you don't have a antibiotic resistance marker, but you have a fluorescent protein. The logic applies always the same. So what you want to do is you count your colonies in this case, and you basically plot the number of colonies according to the dilution series. And what you want to see is a nice linear behavior, right? And that would then tell you, okay, let's say in a dilution of 10 to 6, I have 4 colonies. And that translates then basically to a titer of 4 times to the 10 to the power of 6. Yeah, so that would be 4 million particles per milliliter. That would mean that. And that's important because we need that number later. So now we have a library, we have packaged it into virus. So now let's talk a little bit about the different setups. I don't need to read this, you can listen to me, but 2 definitions we have to make. And we have to make them, I have to make them because they're very, very, very critical for your experiment later on. And the first one is the MOI, which is the multiplicity of infection. And that's already what I said earlier. This value tells us how many viral particles will be ending up in a single cell. Yeah? And for a CRISPR screen, you want to make sure, again, that only one element ends up in your cells. So what we do is with an MOI of 1, every cell would in theory get 1 viral particle. But because this is biology and not mathematics, what happens is that many cells will have 2 or even 3, and some will have 0. Right? You always have a Poisson, in this case it's a Poisson distribution. And so what we want to do in this case is we want to use an MOI value that is below 1. So we want to use more cells than viral particles in the very first experiment. There's a question. You interrupt me, okay?
Okay, sorry, I didn't know if I could interrupt the talk. So I was
please. Yeah.
Just to confirm if it's ideal to determine the titer in the final model that you are going to use then.
Yes, absolutely. That's critical. Oh, sorry about that. That is absolutely critical because the transduction efficiency is context dependent. Yeah, so there are cells that can be transduced very, very well, like for example, let's say primary fibroblasts, for example, or HSF or RPE cells. And then you have other cells that can be transduced very inefficiently. Like, for example, in general speaking, suspension cells. It's much more difficult to transduce suspension cells than adherent cells. So what you want to do is you want to measure the titer in the assay, in the cell line that you're going to use later on.
Okay, thank you so much.
All right, so back to MOI. So you want to use an MOI that is basically below 1. And in a ratio that we find, that we feel comfortable with is something like 0.5. So that means you have double the number of cells than viral particles. All right, and the implications, I'm just here on the right-hand side. So if the MOI is too high, then as said, many cells will end up with multiple guides. And this will confound your phenotype later on, right? But if your MOI is too low, my gosh, you start with a really big experiment of which maybe, let's say 50, 60, 70% later on will die during the selection process. So that you want to find a balance, right? And this, for us, we found the balance with 0.5. But, you know, other labs have different tastes, but we like 0.5.
Is that, do you usually work with confluent cells, sorry?
Excuse me, can you repeat the question?
Do you usually work with adherent cells, sorry? So you're usually working with fibroblasts, because I'm thinking of doing like lymphocytes. So you said it was a bit harder to transport, say.
So a good point. So for suspension cells, I strongly recommend not to go with an antibiotic selection marker, but actually with a fluorescent marker, because it's much more difficult to count colonies with suspension cells. So what you do then is basically you, with the dilution series, you count the percent of positive transfused cells as a means of fluorescence. Yeah, we have done both. So this both works, but in general, our experience is that you need more virus with suspension cells than you would need with adherent cells. So the second definition that I want to make is coverage. So now let's imagine for a moment you're working with a genome-wide library. And let's assume for simplicity, the human genome has 20,000 genes. And we target each gene with 4 guides. That would mean your total diversity of the library is 20,000 times 4, so 80,000. If you were to use each of those guide RNAs only once in your experiment, then there is a very high chance that simply by statistical noise, you're losing some of those elements along the experiment, just because this is biology. So what instead we have started doing is we use each guide RNA multiple times experiment. And that number is called coverage. So let's say if your coverage is 100 times, then each of your elements is represented 100 times. So 80,000 guides in that library times 100, that would be the number of cells you need to begin with, right? Without counting the MOI. We'll come to that. So that's coverage, right? So that's library representation. How much is it represented in your experiment? And classically, in the literature, you will find all types of values, from like 200 to 500 to even to 1,000. And I would say this strongly also depends on 2 things. The first, it depends on the quality of your library. And the second thing this coverage depends on is what is your assay? What is your readout? And we'll come to that later.
Yes, hello, thank you. So I was interested in this MOI thing and the coverage thing. Let's say I started my initial screening with 100 million cells, and I'm using a library that has 76,000 guide RNAs. So
6,000, you say?
Yeah, the Brunello library, let's say, at Gene. So
okay,
when
80,000.
license selection, obviously we'll be estimating, let's say 50% of cells will be lost, right?
If the MOI is 0.5, then yes, yeah.
Yeah, and then let's say from 100 million, I'm left with 50 million. So from that 50 million, should I grow them more, or from that I have to do time 0 and do my downstream applications?
Yes, so that after the selection, or basically from the moment of your transduction, this is your base level of coverage. So in your case, if I just make the math, it's actually quite big. 100 million with the Brunello would be over 1,000-fold. Yeah. So
yeah.
And the Brunello, you absolutely have to do this. That's a different topic. So when you transduce the library, and then after the selection, basically, that would be your sort of like your base number that represents 1,000-fold coverage. right? And from that moment on, your screen starts, basically.
So screen starts from that moment, the initial time point, and not
The screen starts from
the.
so that's a little bit of semantics, because so the way we count is the day of transduction, because when you're selecting your transduced cells, they already proliferate.
Okay.
And while you select your cells, right, you're depleting the cells that don't have virus, they will go down, but the cells that are transduced, they will go up. So very often you see death, but you don't really see much of a change in cell number, right?
Okay.
Because they kind of like they go across each other. Yeah. Again, depends a little bit on the application. If you do a CRISPR knockout, then obviously cells need to repair the damage. So they only start to proliferate, let's say 48 hours after the transduction again. But nevertheless, the numbers will go up.
Because in my case, because when I was doing all the calculation with my team, so after puromycin selection 50 million, and then from 50 million, I have to do my downstream treatments and controls. So it was left as 25 million cells per condition, which is approximately 200 times coverage.
Exactly. And now you are ending up with something that we call a bottleneck problem, right? Because now you're dropping the coverage, and that's actually something that you don't want to do for your samples. In this case, in your case, ideally what we prefer doing is once you have selected yourself, you let them grow for another 2 or 3 divisions, so that you reach the cell numbers you need for all your conditions.
Oh, okay. So
right?
From the puromycin step, I can let them grow more.
Yeah, it depends on the assay, but in principle, yes.
Okay, thank you so much.
All right. So obviously, if the coverage is too low, then you're losing representation, and then just by chance, because biology is happening here, some guides will drop out because these cells, they're not so fit. So that's why we always want to use a certain value of coverage to make sure that this does not happen. So now let's go through a example calculation. We kind of did that already, okay? So let's say we choose an MOI of point, what did I say here? We have it down here. Let's say we choose a 75,000 guide library. We have a coverage of 500. And then we use sort of like an infection rate, which is our MOI in this case of 0.2 or 0.3. It doesn't really matter so much. And the survival through the selection handling, which basically adds up to both of them being 0.5. So you can also simplify this number by just putting the MOI here of 0.5. So then we end up, when you do this math, with roughly 100 million cells. So that's very close to the calculation that we had before, which actually matches the Brunello library, right? And that would be, in this case, a 500-fold coverage. But this is the number you need because of the MOI, right? If you were only to choose how many cells you require for library diversity, it would be half of that, right? But this accounts already for your puromycin selection in this case. So you need a lot of cells. I mean, that's a lot of cells, 100 million cells, right? So phenotypes. Obviously, as I said earlier, they are simple and they are more complex phenotypes. And what I consider more simple phenotypes is basically just plain cell proliferation, right? You have bulk screening, you go in with a pooled library into a dish or multiple dishes, and you just let these cells proliferate. And they can proliferate for, let's say, 10 divisions. That's usually what we do as a minimum, as a rough estimate, is 10 divisions. And that number of divisions depends on the speed of that cell. So that can be translating into 14 days, but it can also translate into 21 days. It really depends on the cycling speed of your cell. But you want to have something in the order of 10 divisions to be able to separate the positive from the negative phenotypes with high confidence. And we'll come to that as well. And this is shown here. So in red, basically, then after 10 divisions, let's say by sequencing and by screening, you have all these genes that increase fitness in this direction. And then the blue genes is basically decreasing fitness in the left direction, right? And this cell proliferation or bulk screening that can be merged now with compounds, co-culture, various different forms, right? But basic readout in this case is cell fitness, is cell proliferation. In the absence, presence of additional conditions, sure, but readout is cell proliferation. Now, in contrast, is are fact-based or cell sorting based. So here, what you do, what you can do is multiple different things, which I would like to highlight. So the first one is shown here. So let's imagine you've done your transaction and you weighted a number of, let's say, divisions, and now you're interested in the expression of a particular surface marker. just as an example. Now you can apply an antibody and stain these, and then you sort based on that antibody being visible by, right? And so you're sorting basically in the ones that have that staining, and you have the ones that don't have that staining. And now you're comparing both populations. And ideally what you want to see, depending on the phenotype that you're interested in and the protein that you stained here, that again, you divide your guide counts into two populations. The one that actually, give you an increased staining signal, and the one that gives you a decreased staining signal. And the same logic can be applied, for example, to a fluorescent reporter for a pathway. Imagine you have a MEP kinase fluorescent reporter, right? And now and the reporter turns green when MEP kinase signaling is on. Then you have two populations at the fax machine, the one that is green and the one that is not green. And what you want to do is you sort both populations, and at the end you compare both and see what's enriched or depleted in one or the other. Yeah, and this can be translated to many different things. So outside with antibodies, of course, or you can turn any sort of like pathway or reporter of your liking into a read. But nevertheless, those are still quite simple readouts because they don't require much sophisticated equipment or handling, despite the fact. Okay, fair enough. So now, more complex readouts are, for example, single-cell sequencing readouts. So that means you're not using the staining of a marker or the fitness of a cell as a readout, but you're actually reading a partial transcriptome of every single cell that you transduced with your guide RNA. Yeah. So again, you have your guide RNAs transduced with your library, and a few days later, you go through one of those machines that basically generates these droplets, merging is tell with some PCR reagents, and then you basically in this droplet you perform an analysis or a sequencing, well, not sequencing, a labeling of the guide RNA together with a transcriptome. So in a single cell, each of these things will get a specific cell barcode, and that can be later then read by sequencing. And this is how you trace sort of like different transcriptomes to a single cell. This is more sophisticated, and it's also way more expensive because obviously you can just not just use regular NGS, you need to do single cell sequencing, right? And also the analysis pipeline is quite different. And so your readout in this case, as said, is not variability or the presence or absence of a fluorescent or marker, but actually gene signature. So the up or down regulation of specific transcriptional programs or individual transcripts even. And the last point I want to make for complex readouts is spatial imaging. And this goes a little bit into optical pooled screens, but that's maybe too advanced for this course at this point. But basically what this means is you're labeling at a specific, a cell that has a specific position, right? And maybe even additional markers within this. So you're using not a fax or NGS, but you're using imaging like a microscope, to visualize your cell. At the same time, you're visualizing the guide RNA that is in a cell. And so now obviously everything that you can image can be used as a readout. So this can be a protein localization. It can be the abundance of a protein. It can be the morphology of it. It can be even the transcriptome of a single cell. So this is certainly the most advanced and complex phenotype that for pooled screenings that you can use at the moment. So now let's go through an experimental checklist. And I divided this a little bit into what you want to make sure before and also what you want to make sure after. So we want to make sure that we have a good library and that we have a good virus prep, right? So that means plasmid library representation, and we want to make sure we have good titer, right? High titer. You don't want to work with a virus of low titer. You want a high titer because otherwise you're basically just pouring media supernated on your cells, which may have an effect independent of the virus. The second thing is cell line readiness. One of the things that we over and over see, make sure if you're working with an engineered system that your Cas, your nuclease, is actually active in your model. And I cannot emphasize this enough. I mean, how many screens have we done from collaborators realizing that the Cas was not active? And that's always a bummer because you go through this long process and then you don't see any phenotypes. So test that before you do the screen, right? if you are using an engineered system. Of course, there are also libraries in which they are all in one libraries that deliver also the Cas9, but then you have other issues with that. So that's absolutely critical. Transaction and selection, that's something that we touched upon with the question before a little bit. So you want to make sure of two things. The first one is what we call the bottleneck tracking. So you want to make sure that you always work with sufficient coverage of your library. But the second one is also the population doubling. So that's phenotype and assay execution. And I give you one example that is a plot. When somebody in my lab does a screen, I want to see this plot no matter what. And that's not even sequencing. And it looks like this. So that's just an example. So what this shows you here is in blue is the library representation at each split, basically. And what you see, we start with a certain library. We start with 100-fold coverage. And then during the first couple of days, it goes up to maybe, I don't know, 500 or something, right? And at the same time, we plot here, and that's a log2-fold calculation of the cell numbers. So at each split, you count the cells, and you want to know how many doublings of the cell have. And this is shown here in red. And you do this for each split. And in this case, we had two splits. Yeah, and you see the doublings, they go up quite a bit. And then you end up with, I don't know, in this case, 7. And then your coverage also goes up. And then you can accumulate sort of like, what is the accumulated coverage? Although this is sort of like a number we can debate, but the doublings are more important. And what you want to see is that I did the library representation as well as the doublings, that they constantly go up, that you don't have hiccups in your screen, that the cells don't behave differently during the different splits. So that's something I think that is very critical. And that avoids bottlenecking. So you make sure that you always have the sufficient coverage in your experiment in the blue line, but also that you have sufficient cell doublings to actually see population separation later on when you do quality control. So the last thing that I want to point out on the checklist, second last thing on the checklist is optimize your PCR conditions before you do the screen. So because you have to do the PCR to sequence your guides later on, and you want to make sure that you have really well working conditions here. And the point I want to make here at this point is be aware of template switching. And that's particularly PCR template switching. And that's particularly relevant, not so much for libraries that have only a single guide RNA. But the moment you're working with libraries that have multiple guide RNAs, this problem, template switching, becomes really, really, really important. And so the way this works, just briefly, don't need to go through this thing, but imagine you're doing a PCR and your polymerase, by whatever means, in the time that you give the PCR, is not capable of fully elongating the template. So what happens then is that you end up with a partial product, a partial PCR product that is not full length. So up until this point, no problem. But when you now go through a denaturation step, that partial product becomes the perfect primer for the next cycle. And if you have two elements in your PCR product, let's say you have a guide RNA in this position, and you have another guide RNA in this position, the moment this is not fully amplified, this primer binds to a completely new strand, and you decouple the two guide RNAs. So you end up with combinations that you did not intend. And that can be a really, really big problem for combinatorial libraries. So you want to make sure that actually you're using very, very few cycles of PCR, as little material as possible, and so on and so on. So yeah, so that's the point I wanted to make here. So optimize this, and maybe also go through just a quick and dirty nano kit sequencing, just to make sure that these template switching rates are really little for QT. And then at the end, I mean, this is more like to make sure you're working with the right cells and so on. Sometimes cell identity. I mean, sometimes you see people working with cells that have been used in the lab for, I don't know, a decade or longer. And then the question always comes, are you sure that those actually are the cells that you think they are? Because, different people, different stocks, and so on and so on. So again, you want to make sure that you're working with the right thing, right? And also contamination checks. And that goes back to the first, I think here. Yeah, so mycoplasma is a huge problem. So just make sure that this is all clean before you start. So this is sort of like the, I would say, the experimental part. Now we come to the last two parts, which is NGS and then also a little bit on GNM. So I dropped here part of a plasmid that we routinely use. And what you see here is that you have this RNA promoter, in this case, is a 7SK promoter. And it drives, in this case, imagine this is a placeholder here, but at this position would be the guide RNA. And then right adjacent to the guide RNA, you have this GTT, and that's basically when the scaffold starts. So that's the part that binds this line. So now, for PCR, when you have a single guide, ideally what we would take, we would design a primer that somewhat binds in this region here, yeah, and another one that binds in this region. So you end up with something like a PCR product that is shown here. Yeah, so you have NGS adapters for Illumina on the 5' as well as on the 3'. And then you have a tag or barcode to make sure, you know, that you can separate samples. And then there are two modes that you can sequence this. And some of you may know this, some may don't. So you can have a single read. So that means you're just sequencing in one direction as long as you can or need, or you can do a paired end sequencing in which you sequence a few cycles in this direction and then in a few reads in the other direction, right? And then ideally you want to make sure that in the overlap you have your guide RNA or even two guide RNAs at the same time. And then that is basically happening on the machine. So now you get these nice FastQ files. What are you going to do with those, right? Because you need to generate read counts. And I just want a little bit of a self-promotion here. So that's a tool that we developed called Rico, just for read counting. And so GitHub, you can use it. And it processes basically single reads as well as paired end. And you apply, so what the software does, it recognizes automatically where your guide RNA is positioned. You can also tell it, but it recognizes it automatically. And then the only thing that you need to provide is your library file. So basically a CSV or an Excel file in which you have the guide RNA name and the guide RNA sequence. And then the rest, the software will do automatically. It will recognize the reads from the FastQ file. It will do the trimming. It will do the cut adapting, as well as then the counting and aligning to the library. And then you end up with a table that is shown here, like guide 1 is maybe once, and then RNA, what is it, 34 is found 7 times, or the third one is 59, and so on. And if you have two reads, it works similarly. It just goes through both reads simultaneously, and then you get an output basically for both of these guides. So basically combination of 1 with 12 is found 20 times, 22 times, and 7 and 35 is found 70 times, and so on. And that the output is shown here on the right-hand side. So, and what I said earlier, for trimming, alignment, and also the template switching rates, this is shown here as an output graph in blue or in red. And in this case, what we always like to see is that these two bars are very low. That means basically that your alignment rate, so the reads that you got out of this sample, in this case is more than 90%, they could correctly map to your sample, to your library file. And that's what you see. So that tells you PCR was good, and I mapped the right sample. Then you also see sort of like your sequencing depth. So in this case, it's very lowly sequenced, actually only 24 times, but I show this for a reason. So it's very, very shallow sequence. Then you see a sigmoidal plot of the sample. Yeah. You also see a Lorentz plot on this case. So that means this is sort of like the cumulative frequency of these guides, so in percent values. And you have a histogram which shows you the skew. And the point I want to make in this case here is, even with a library where the sequencing depth of only 24, we basically see 99% of this genome-wide library. So that already tells you that it's extremely uniform, right? Because you don't need to sequence deep in order to see everything. But this comes later on. And on the left side, I'm just showing the time the software takes for also complex library, depending on the number of reads that you generate. Yeah. So in most cases, we now tend to use like NovaSeq reads with way over 1 billion reads. And the last thing you want to do is wait hours for the read counting to continue. Yeah, you want to wait half an hour or a few minutes, and then it should be done. And that just shows you just how read counting scales with respect to the size of the data with software compared to Pineapple Pi. Yeah. All right. Now let's talk a little bit about QC. So string QC. The first thing that we do is we check read counts and ask the question, have we sufficiently sequenced? So if I show this plot to you, which shows you a couple of sample names on the left side, and then just some histograms or box plots, in this case, of the normalized read counts log 10. So that means, in this case, 100 times. Now, my question to you is, if somebody shows this plot to you, can you answer the question, have you sufficiently sequenced your sample? In this case, I would say, no, you cannot, right? This is way too simple, okay? Just by looking at how these reads are distributed, you will not know whether you have sequenced deep enough. Another way of visualizing is this. You can do a histogram, same sample, it's just one of those samples. And now you see it's sequenced, obviously you see a nice histogram, right? You have some low read counts, you have some high read counts, and then the bulk is in between. Can you tell from this plot whether you have sequenced deep enough? No, you cannot. Because there are still some effects down here that you cannot control for, right? See how this population goes further on in this direction. See, you're cutting it off right here. So obviously there's a tail. And you don't know actually how far the tail goes. So in order for your analysis to be accurate, you have to process these data, or you sequence even deeper. But in this case, I would suggest to process them. And the way we do this is shown on this graph. So that's a very complicated thing, but at the end, it's very easy. So what I can explain to you what we do here. So on the x-axis, you see the read counts of all your elements, and they are binned into bins of, I don't know what it is now, 50, I think, right? Yeah, 50. They're binned into groups of 50. And on the y-axis, you have the log fold of every combination with your guide RNAs. And now what you can do is, because you have the log fold of that combination, you can ask, what is the resolution that I have on my high counts, on those guys, and on my low counts? And you can immediately see that something is fishy up here, right? Immediately, they don't match the profile. You have a very stable correlation, that is centered around slightly negative amongst the read count starting from 50 on. But before, it doesn't look good. It really doesn't look fine, right? So ideally, in this case, what you want to do, you draw a line and you cut the data off. And in this case, it's 1%. And the point that I want to make is that in this case, it's a sample from us and it's 1%, but that can vary and depend on your sample. So you want to do this type of analysis because sometimes this line is maybe here. And it can be there because you haven't sequenced deep enough. And it's a very, very easy mistake because if you have low read counts, a change from 1 to 2 read counts is already a log fold of 1. And depending on the phenotype you're scoring, a log fold of 1 can be enormous, can be really big. So you're prone to a lot of false positives in your analysis just because you haven't sequenced deep enough. So bear this in mind. That's something that you want to do. Now, the next thing that we generally do is we look at correlations of replicates. Obviously, you want to have replicates, not only technical replicates, but also biological replicates, right? And in this case, we're comparing biological replicates, replicate 1 versus replicate 2. But the point I want to make is you do this not only on the normalized read counts level per guide RNA. No, you also do this on the gene level. So you're aggregating the normalized guide counts on the aggregated gene level, as well as on the gene log fold level. And you want to see a sort of like increasing correlation between guide and gene level, right? And you want to see a very decent correlation between the phenotype, because this is normalized read count. That just tells you how similar is my sequencing. It doesn't tell you anything about the phenotypes that you observed in your experiment. Only the log fold, that will tell you something about the phenotypes. So this tells you, oh, my sequencing was okay. But this tells you whether my assay is actually reproducible. Right? And keep that in mind, it's very important. So we like to show all of these things as QC metrics. All right, next thing. Now we know replicates correlate and phenotypes correlate. The next thing we want to make sure is basic QC. So we have a certain expectation on certain phenotypes, and we can ask whether those match our observed phenotype. And that's the plot that I'm showing from somebody in the audience. So, and what you see here is, again, it's the same graph that I showed you here on the right-hand side, right? But the only difference now that we highlighted a couple of populations of different genes. And in this case, we highlighted the core essential genes that I mentioned in the very first slide, you remember, right? The very first experiment. Those are the genes that are essential for cell fitness. Those are highlighted here in red. Then we're highlighting in blue the genes that we know that are basically never essentials. Right? You can deplete them because they're not expressed or whatever. Then we have some other control sequences that don't have a target sequence in the human genome, as well as a control sequence that is a safe harbor load. And what you want to see is that these populations separate well from each other. That's what you want to see. But seeing is not enough. You need to quantify. And the way to quantify this is quite simple. It's called Cohen's t-statistic. Many people get stressed out when they hear statistics. It's a very simple calculation. And basically what it asks is how far is sort of like is my red population in this case, the red one here, how far is this one away from, in this case, sorry for the color code, from my blue population. So the gray in this case is the blue. Yeah. So what is the distance between this in terms of log folds? And you can also do this with the NHTs or with tumor suppressors on the positive side. So you're asking how far are the essentials away from my controls or the tumor suppressor from my controls, right? And a good screen not only starts with a good library, but it also has a good Cohen's d value. So a value that is way above 1, at least for the essentials. I mean, like square. So basically 1, -1 or +1, it doesn't matter, right? So you want to have a value above 1. So then you have quantified it. And if this looks decent, all right, now you know your controls behaved well, your assay behaved well. Now you can go the next step and say either data analysis, or as I've shown here, you do yet another additional analysis, which you can do, but you don't have to do. So what we've shown here on the left-hand side is the dropout of essential genes, right? You can also ask more generally, how good is the data set in finding those essential genes? Yeah, if you just use viability as a readout. And this is shown here by this ROC analysis on the left side. So this tells you basically if the value is close to 1, then the data set is perfect in finding essential genes. And then on the right-hand side is the precision recall in this case. So that asks how certain are you that the genes that you found to be essential are actually essential. So this takes also the true positive and true negatives into account. And then ideally what you want to see is with a genome-wide library in a proliferation screen is that both of these numbers here, that they really close to 1. So basically 991 is by far the best screen that we've done, but it's also because of this library that it's unpublished, but it's available from Biblion. It's a genome-wide library. So it really, really works extremely well. Okay, now, there is one more thing that I want to say about QC. And you can make the analysis of whether your coverage was high enough in your experiment. And the way you can do this is, imagine you've done a screen in 2 different coverages. And you're taking the higher coverage, the number of essential genes that you identified in your higher coverage as a baseline, and you ask how many of those do I find at a lower coverage? What you want to see is that this line, according to the FDR threshold, is a horizontal line. So the line should not change, because regardless on whether you screen it at a higher coverage or lower coverage, the confidence that you have that a dropout in an essential gene is essential should stay the same, right? So for Alexandria, I want to make the point that it is a horizontal line, regardless on whether you screen this thing at 100 or 200-fold or 50-fold. And remember, we've made the calculation for Brunello earlier for 1,000. So this is a genome-wide library. You screen at 50-fold, and you don't see a difference in hit confidence compared to the same library at 250. Whereas the second best example out there in the literature, the TKO library, you see how this line goes up and even dramatically here. So then you can make the argument, these libraries have not screened with efficient coverage. So I would have needed more cells actually to be confident that my dropouts are dropouts. So that's next level analysis. Okay, I will skip this just because. And this was for, so the point I want to conclude on this here is that a library with a lower distribution, so more uniform libraries, they can be screened at lower coverage. That's the point I want to make, yeah? And that's important because, you know, you can use sparse cell material, primary cells, you can go in vivo, single cell readouts. You know, if you do genome-wide library with a non-uniform library, you need to sequence 1,000 or 500 cells for each guide RNA, which is crazy expensive. But if you only need to sequence 5 or 10 to get the same information, that's a huge saving. And obviously combinatorial, right? This is where it really starts to matter because numbers just go through the roof. All right, hit calling. Very briefly, I know you touched upon this already. There are so many tools out there, and I don't think that even an hour would be sufficient to go through all the tools to explain their pros and cons, and also for which applications they are good and for which they are not so good, and so on. So I really have to cut this, excuse me, but if you have questions along the analysis, please reach out. Happy to answer these questions, yeah? So what I focused on was actually magic. I know you've done this already. I think it's a good start. It is a good start. It is not an ideal tool, let's be honest, but it is a good start for any type of screen that you've done, just to make sure that your control is behaving well. And I know that you've done this, so I can skip this really quickly. So you get these two files, right? You get the guide summary and you get the gene summary. And ideally, right, you jump right into the gene summary. You check, are the essentials gone? Are the tumor suppressors up? How many guides are actually associated with the phenotype? And then you get all these nice plots. And obviously, you want to see the same, right? Okay, great. Now, I have a few more slides. I know we're out of time. You know, taking all of this into consideration, what I just told you, uniform libraries, and you can downscale experiments. What we've been doing is we generated a genome-wide library that has only 40,000 elements, and we call it Pergamon. And what we did is we did a combinatorial library with this genome-wide and targeted roughly 27 genes along these 40,000. So you have a matrix in which you deplete 27 genes in one dimension, but genome-wide in the other, and all of them in the same experiment. So that's basically 27 genome-wide screens in one. And this is just the status. And what I want to show you is that when you merge this with essential genes, then you basically skew all the Pergamon phenotypes to the negative side. So that looks really good. And if you merge this with a tumor suppressor, then all the genes become, you know, more enriched, more positive. And then, of course, some non-essential genes, they are somewhat in the middle. And now what we can do with this data set, and what I'm showing you here up here, sorry, is this Cohen's p-value, right? So how far are these populations away from each other? In this case, right, it's 3 something, and in this case, the average is 2 something. So it's a really good screen, even though it's like humongous in its scale. And so what we can do with this, obviously this is QC, I will spare this, we can ask whether we see unexpected phenotypes, so whether we can see genetic interaction. Because one of the things that we have in this library, just pointing this out here, are the CCNL1, CCNL2. Those are non-parallel genes. So they don't have a phenotype on their own. So whenever we expect genetic interactions to occur, this should pop up, right? Just skipping though right here. And among the top ones that we see is basically the parallel genes, right? And so you can see that this really works well. So that means the single depletion doesn't do anything, but the combinatorial depletion really does something, and we can score this. And we not only see negative ones, which are synthetic lethals in this case, but we also see very positive ones, right? And I think this is really cool. And we can actually do this in not only one cell line, but we can do this in multiple cell lines. So this is wild type cells, and these are replication cells. We can correlate these two and then ask, what patterns do we see? And one really interesting thing that pops out here is all these yellow dots, this one, this one, this one, this one, this one, this one, this one. So those are all gene pairs with a gene that is called CYP2A. And so that actually confers, that identifies CYP2A as a genetic interaction with replication stress. And now CYP2A, one should know, is a DNA damage associated gene. And it's actually very revealing because it's a known synthetic lethal pair from the literature. So just showing you some snippets on what can be done with combinatorial genetics. And if somebody's interested, then please, yeah, reach out. All right, I summary and I'm done. So first, the concept of individual steps of full CRISPR screening. I hope that I delivered this. Avoid gray readouts. aim for black and white. That means binary readouts, yes or no answer. Everything gray makes things complicated, right? So really the phenotype matters a lot. Screening checklist ensures that all steps are well taken care of. I cannot recommend this enough. Don't just use the NGS data. Make sure they're of sufficient quality. I think that's also something that I hope I deliver. And don't jump to hit calling, right? Make sure that your screen is actually of sufficient quality, with all the controls that we mentioned. And then through the hit calling pipeline, there was insufficient time today, I give you that, but established for your readout. So for example, Magic great for viability, but when you do drug screens, for example, I would rather use DrugZ, right? Or maybe even Bagel or Bagel 2. So there are different tools that depending on the application. And the point, obviously, I want to make uniform libraries allow downscaling to be cost efficient. Yeah, and with that, thanks a lot for your attention. I know I exhausted my time, but I hope you learned something. So happy to answer any questions if there's time left. Thank you. Do you offer end-to-end CRISPR screening service, including analysis support? So the partner included customer service. So yes and yes and yes. We're partnering with, yeah, we do tailored libraries, we do off the shelf. Yeah, I think this sounds all very reasonable. I don't know who was that person, but yeah.
But we want you to give it a go yourself first.
No.
Right?
One thing I want to mention though, however, is because I talked about this Alexandria library, and it's not yet published, but we're really working on this. And it is as cost efficient as if you were to go to Etchin and buy any other library. Yeah, but it's for academics at least. So in this case, reach out to Vivlion. They're happy to send it over to you. Works amazing.
Yeah, I think the point it was to really, have these conversations, Manuel, and kind of really expose everyone to what's available out there. Different things will fit different people, but we just want to make sure that we really empower people to access CRISPR technologies and CRISPR things as much as possible. So really, we appreciate sharing your story and your tools.
Anytime, Hannes, anytime.